I said I'd talk about this for about 15 minutes, and I looked at the schedule and I said, what, I've got half an hour? Okay, I better do something serious about here. So the subtitle of my talk is Of Mice and Men and Fruit Flies. So you see the mice, the male model, John, and the fruit fly. So I'm going to talk about how researchers use different models, different cells, different animals, and even studies in men to try and understand how Kennedy's disease works. And of course we hope by understanding the basic causes of Kennedy's disease that we'll be able to find a solution, that we'll be able to find something that will help the symptoms. Okay, so this is my model of a cell. This is a very, very basic model where the cell is a circle and it has two parts, the cytoplasm and the nucleus. And you could think of a cell, in some cases, about like an egg. The cytoplasm is sort of the white of the egg, and the nucleus is the yellow yolk. So the androgen receptor, which you know it's the faulty androgen receptor, that causes Kennedy's disease. Usually it's in the cytoplasm of the cell. But when it binds to testosterone, a male sex hormone, or dihydrotestosterone, the androgen receptor goes into the nucleus and then binds to DNA. It's a lot more complicated than that, but we'll start with the basic model. So here's my little animation of the androgen receptor in the cytoplasm of the cell. In this case, the androgen receptor is red, because sometimes we can color the androgen receptor red or green, depending on what sort of microscopes we use to look at it. And then we can add testosterone or dihydrotestosterone to the cells. And then actually, in real time, about 4 hours or so, the androgen receptor can go to the nucleus of the cell. And one of the cell types that's affected by Kennedy's disease is the neuron. And I'm going to be quoting some papers throughout this presentation from many different groups. Of course, I don't have time to go into all the models and all the results. So I'm just picking a few from general models. This is work done by Angelo Paletti's group. And I understand he may be here at the conference later, maybe?
So, yeah.
I think so, that's good. So I just wanted to show you a picture of a basic neuron. It's very different from the cell I showed you before, which looked like a fried egg. This one is very extended, it's long, so you can imagine the neuron is in the spinal cord, and it goes all the way down and then goes out and attaches to a muscle. So for a long time it was thought that it was the neurons were a problem in Kennedy's disease. And it is true that the neurons do degenerate or die in Kennedy's disease. So Dr. Paletti's group used model neuron cells to check out where the androgen receptor was and what happened when you added testosterone. Now another thing about the androgen receptor is normally it has about 20 glutamines. Now glutamine is an amino acid. Proteins are made out of amino acids. Now there's a language in biology that amino acids each have a different one-letter code. Now it happens that the code for glutamine is Q. The G was already taken up by glycine, so they had to give glutamine a Q. So an androgen receptor with 22Q, is about normal, whereas one in this case, shown with 48Q, is abnormally long. And that's what patients with Kennedy's disease have. They have an androgen receptor with more glutamine amino acids than usual. And that's sometimes called a poly-Q tract in the androgen receptor. So the more poly-Qs you have, if it's above 40, that results in Kennedy's disease. So when Dr. Paletti took these neuron cells and added testosterone, I'm going to use my finger, they see the androgen receptor went to the nucleus of the cells. In the cells with the poly-Q expanded androgen receptor, it did go to the nucleus, but there are also these aggregates formed in the neurites, which was very different from the normal situation. And aggregates are, in this case, they're not nuclear inclusions, but were thought maybe to be a problem in Kennedy's disease. Now there's some debate as to whether they're actually a problem or they protect the cell by putting the androgen receptor in a place where it can't harm the cell. But this is one of the models. This is some work done by, I guess, Dr. Chang's group with Diane Mary. And you may have heard about ASCJ9. Ameliorates, makes better. Scientists like to use fancy words. Spinal bulbar muscular atrophy by degradation of androgen receptor. So we're saying that this drug makes Kennedy's disease phenotype, or the appearance, better by making the androgen receptor go away. So that's the translation of that. So these cells have the androgen receptor with 112 glutamines. And when the dihydrotestosterone was added, they formed aggregates in the cell. And when the cells were treated with ASCJ9, you see less androgen receptor, or it's throughout the nucleus of the cell. So this is another model in cells to say, okay, what possible treatments could we use? We'll start with a cell model to see the effect. Now, another thing that we've studied in cells is proteasome activity. And you may have heard me speak about proteasomes before as a sort of the recycling bins of the cells. And in this cell model, we studied it using a green fluorescent protein. So these are really cool. You can look at cells, and if the proteasome doesn't work, the green fluorescent protein hangs around and the cells turn green. And you can look at this under a microscope. Now, when we did some of these studies, these are the cells that just have the green fluorescent protein, faint green. If you add a proteasome inhibitor, they're very bright green because the green fluorescent protein can't be degraded. And in these cells, with the normal androgen receptor, you can see the MB is a synthetic androgen that we use in the lab. The androgen receptor is in the nucleus, the green protein disappears, and you just, when the cell slides are put together, you just see the androgen receptor. Now, with the androgen receptor with more glutamines, in the absence of the ballerone, it's in the nucleus, or not quite in the nucleus, you don't see much green fluorescent protein, and when it's merged, it's still red. When you add the hormone, the androgen receptor is in the nucleus, the green fluorescent protein shows up strongly, which to us indicates that there's somehow the proteasome isn't working. The recycling of proteins isn't going on well in the cells, so that's why you get a green cell. And if you merge it together, it's like some of those signs you see, maybe in a subway car, with the red and green lights together make a yellow light. Well, that's why we get a sort of yellow-looking cell there.
Lenore,
Yep.
there is a pointer in Mike's case. Would you like to have that?
Oh, sure. That works. Yeah, this one is.
Yeah, it's kind of dim.
Yeah.
We got a little brighter pointer.
Make it easier for you.
Yeah.